Hello friends. Today I'm going to discuss about your translation and the process which involved is your initiation. And in the coming slide, I will be discussing about your elongation and termination. So, what is your translation? You can see in the figure that I have shown this central dogma that is from DNA. DNA makes its own replication, own copy by the process of replication, and through the process of your transcription, messenger RNA is produced, and through the from messenger RNA, your translation is protein is produced through the process of your translation. So, we are going to discuss about how this translation take place. Actually, translation it is a mechanism of protein synthesis from mRNA templates. So the mRNA are in fact the blueprints of DNA where the genetic information is encoded in alphabets, which get translated into protein. So synthesis of your peptide bonds is the principal chemical events in protein synthesis. So in 1950, Paul Jemnik he first showed cell-free protein synthesis in the rat liver extracts. And then he also showed the requirement of your ATP, GTP, and also he showed that ribosome as the site of your protein synthesis. So we know that I have already said that translation is the mechanism of protein synthesis from mRNA templates. So what are the different pigments of your color? It is shown here. That is your skin color, hair color, and your eye color. So this skin color, hair color, and eye color, how this is governed? So yes, our DNA has the genetic information stored in it for the pigment which is responsible for this eye color, hair color, and skin color. And what is this gene? Gene is a small segment in the DNA. So there are a portion of DNA that codes for some proteins, and this protein makes the pigment responsible for your eye color. So next, prokaryotic mRNA and eukaryotic mRNA. So in prokaryotic mRNA, you can see this has multiple translational start site, and it will also be having your stop codon. So here suppose the AU is there and the translation starts, will get one protein, again there will stop codon, and then again another protein translation if it starts, again another AUG will be there, and so on. It has multiple translational start sites, whereas in eukaryotic we are having only single translational start site. And as eukaryotic is a very large, here after post-transcriptional modification only your translation starts, whereas in eukaryote after transcription has not finished also your translation starts. So this is the difference between the two. So in this we're going to learn that what are the components we require for it, how amino acid get activity, and then how this protein synthesis take place. So first we are going to learn this protein is the first recommendable component. As we know that from by making your factory raw materials are required, in the same way your amino acids is required as a raw material for making your protein. So and next is your ribosome. Ribosome is the site of your protein synthesis, and it gets associated with a rough endoplasmic reticulum to form cluster RER ribosomes where protein synthesis occurs. Then we need mRNA. As I have said already, DNA has passed the genetic information through the process of transcription in the form of codons to mRNA to transcribe into a protein synthesis, which will be happen by a translation. And what are these codons? Codon is a trinucleotide sequence of DNA or RNA that correspond to your specific amino acids. Then we need your tRNA. What is your tRNA? tRNA is having, it looks like your clover leaf, and it is having your 4 arm, that is the receptor arm, that is CCA, to which your amino acids will come and bind at the 3' end, and it will be having your T psi arm, which is 5 base pair. Again it will having D arm, which is 4 base pair, and this is anticodon arm. So what is this anticodon arm? So in the mRNA we are having codons. Suppose if it is AUG is the codon which is for your methionine, then this tRNA will come and binds your, and it will having the base UAC, which will base pair with it in this way, UAC AUG. This is complementary base pairing, and that's why it is called anticodon, and this is the codon. And also this tRNA is having a variable arm, and based on this variability it has also classified further, but this I'm not going to discuss here. Now another is energy source. For doing anything we need energy. So both ATP and GTP is required for the supply of energy in protein synthesis. So for activation of amino acid has to occur before it can become attached to tRNA adapter. So therefore for this energy is required, which is produced by cleavage of your ATP. And GTP is required for one molecule of GTP is hydrolyzed in the release of EFTU GTP complex in prokaryotes and also in the process of eukaryote. So EFU means elongation factor, which will be discussed in the elongation slides. So again we need protein factors. So protein factors are tangentially associated with ribosomes for taking this translation process to occur, and there are different types of these protein factors are there, and they are named as initiation factors, elongation factors, and termination factors. So here in the initiation factors it has been named as IF1, 2 and 3 in case of prokaryote. And in case of eukaryotes, the prefix E is used. Therefore, it is supposed IF instead there will be EIF1, 2, 3, 5B, 4, and they have different functions like they block excite entry of initiated tRNA and blocks reassociation of two subunits. So then how this amino acid has to be activated, how it will get activated. So amino acid come and associate with your amino cell tRNA synthesis, and with the utilization of energy from ATP, it forms a complex which is known as EAMP amino acid complex, and then it comes and bind at the 3' end of the tRNA and form the way forming your amino cell tRNA. Then as I've said already that there are 3 stages of protein synthesis, that is initiation, elongation and termination. So protein synthesis take place in ribosomes. So ribosomes are having your 2 subunit, that is in case of prokaryote it is 50 and 30th subunit, whereas in eukaryote it is 60 and 40th subunit. And all the ribosomes have their 2 common sites, that is P site and A site. A site is a site where amino cell tRNA comes and binds. It is having anticodon and codons in the mRNA, which I have discussed. And then this is your binding site for mRNA. Then what happens in prokaryotes, the messenger transcript has a SD sequence, which is known as signed Dalgarno sequence, and this sequence is recognized by a complementary sequence which is present in 60S rest ribosomal RNA, and then they base pair with each other, and then they proceed towards the start codon AUG and translation starts. Whereas in eukaryotes, ribosome small subunit recognize and bind to messenger 5' cap, and therefore and then they recognizes AUG, that is start codon, and starts translation starts. So here you can see this is the codon segment. It is stop codon. The stop codon can be your UAA, UAG or UGA, and then your termination take place, and at eukaryotes there will be poly A tail 3' N. Sometimes AUG identification is facilitated by a specific sequence of nucleotides surrounding it. It is called Kozar consensus sequences. Now, in prokaryotes, the initiating amino acid is N-formylmethionine. Yes, formyl group has been added to this amino group. Therefore, it is N-formylmethionine. Whereas in eukaryote, this formyl group is not attached. Only that is the difference. Next, how this initiation of your translation will take place. So some initiation factors are there, like IF1, IF2 and IF3 and GTP are required. So first, in the first step, you'll see here in the figure that this is 70S ribosomes. It gets dissociated into 50S and 30S. Now IF3 comes and bind to 30S, so that this 30S don't go and reassociate with your 50S. Then this messenger IF1 binds to the small subunit, and this IF1 it assists the charge initiator tRNA and mRNA to bind this complex, and as IF3 get released. So this IF2 recognizes this F methylene tRNA, and then the release of this IF3 here, IF3 get released, and then 50S comes and reassociate with 30S subunit. GTP is bound to the IF2. Here GTP you have seen here, it is bound to the IF2. It gets hydrolyzed at these steps on 50S entry, on this 50S entry, and release of this FIF and IF2, they two get released. Finally your 70S initiation complex is formed. Next, in eukaryotes, the difference is that it is having at least 9 initiation factors are there, and a prefix E is used for them. And as I've said already, the ribosome sedimentation coefficient is of 80S in eukaryotes, that's 60 and 40S. So what happened here is here 5 different stages are there. First, a ribosomal dissociation will take place, as in case of prokaryotes, your 60S and 40S subunits are dissociated. Now initiation factor 3 and I1 comes and bind to this 40S. So when this acts like initiation factor IF3, as in case of prokaryotes, it will not go and reassociate with your 60S. Now next step is your formation of initiation complex. For this EIF2, which is a GTP binding protein, here GTP is associated, it brings this initiator tRNA and to this your small subunit is 40S subunit. So the messenger RNA 40S subunit with the help of your EIF4 and EIF3 and cap binding protein, all they get associated. So you can see here this is a ternary complex. It is formed in presence of your initiation factor 2B, GTP and 2C, and this ternary complex comes and bind to the small 40S subunit, and they were forming a 43 pre-initiation complex. So how this ternary complex is formed? So this ternary complex, here I have shown that finally formation of 80S complex, what happens at the end, all the initiation factors get released and they again recycle for again repeating the same thing for making again that 80S complex. So in that case the activation of then EIF2, the head has become now deactive. For activation of EIF2 it requires EIF2B, and which is also this EIF2B, and this EIF2B is also known as guanine nucleotide exchange factor. So it activates EIF2, and then this activated EIF2, which is bound to your GTP, it requires EIF2C to form a ternary complex with your methionine. And then thereby a ternary complex containing your methionine tRNA and EF2 bound to GTP, it attaches to your 40S, as I have shown you in previous slides. Ribosome subunit to form your 43 initiation complex. Next, then 48 initiation complex is formed. Now how that is formed? So as the ribosomes in prokaryotes is having 5' cap and 3' poly A tail. So what happens, the different initiation factor like 4G and 4A, it get associated with 4E, and therefore a 4F complex is formed, which is known as your cap binding protein. And this is called cap binding protein because it goes and attaches the 5' cap region of the mRNA. Then your ATP is hydrolyzed by your EIF4, which drives this 40S initiation complex along the start AUG codon. So you can see this EIF1 4F complex, it gets associated with 43S complex, which I have discussed already. And then you can see here, after that this ATP it get hydrolyzed, and this for 40S initiation complex now along with the start codon, it is now ready. This is again, you can see here IF4 get again released. Only this complex IF4, 5, 3, 4 and 2, that is 3A2 is there in the small subunit along with your mRNA and tRNA. Now what happens now, another initiation factor 5, it induces and releases IF2 3A, and this IF5, what does it do? It triggers the hydrolysis of your GTP bound to IF2, and it results in the release of IF2 GDP plus other initiation factor. And finally, your what happens? 67 joins with your the small subunit forming ATS initiation complex to form this active S ribosomes. And now it is now ready to go in the next step, that is elongation step, which I will be discussing in my next class. So friends, there are lots to learn. It's a very vast and interesting topic. If you want to learn, go deep, you read more and more books and subscribe my channel if you like my videos. That is bio class for you. Thank you for watching my videos.